RNA, and the red means actually a successfully folded protein, is just really cool. So to remind you all the central dogma, of course, it's the guiding principle that DNA gets coded into RNA through transcription, which then is translated into amino acid chain, which become a protein. And that, of course, the information only flows in one direction from DNA to protein. It doesn't go backwards.
And just so you guys know, I'm sorry,
Go
just
ahead.
so you guys know too, I'm not sure what kind of sharing abilities we have given you at this point, but I loaded a ton of stuff into the Google Drive that has notes that are editable for you guys to, you know, build up or par down for central dogma with the specific steps of DNA to RNA and RNA to protein and transcription and translation. Sorry.
Oh, you're good. I'm a fan. I am
You're
dying.
dying.
It is so hot
It's
in my
really
room.
hot.
I feel like I'm in this, you know, Central Africa. So anyway, of course, students, I find, I'm dyslexic, so I found it very difficult when I was learning this to understand transcription versus translation. They both begin with trans. And so I always talk about to the students about, you know, transcription is transcribing, which means you're just putting it into another language. And then translation is fully, or excuse me, transcription is you're writing it down, right? You're transcribing it, you're just writing it. And then translation is fully going into another language. And so, no, you can't unplug it. Okay.
You can't have a fan.
Okay, no, how about there? Anywhere, I just need a fan anywhere. Sorry about that. So I found that's an easy way for kids to kind of grab onto the idea that transcription and translation. Do any of you have a clever way that you get your kids to hook in on that and don't get confused?
I put out like the C and the S in the word, and I will tell them, like, you know, you're just, the C comes before her. So I'm already confused myself. C comes before L. And so that step is first and the other step is second.
Oh.
And so in the alphabet, because it's, as much as I try to explain it like you just did, they always like,
Yeah.
they never get it. So I'm like, just which comes first in the alphabet? Transcription is first, translation is second. And then
I love that.
they seem to get it. That helps.
Anybody else?
I always use it like a language. I say, you know, it's a language with 3-letter words. All the words are 3 letters, these codons, once we get to that. So I'm translating it from this code to the language, right? To the words are all 3 letters. So that's how I do it. And they always remember translate. Okay, I get that, translate from a foreign language. Well, that's how I do it. And the transcription just is the other thing. It's the precursor to that, I guess. I compare transcription to, you know, replication, because it looks just like it, except it's RNA nucleotide.
Yeah.
So
That's another good comparison.
So I tell them everywhere, it's like replication, except everywhere you think you should put a thymine, I'm like, what should you put, guys? And
right, put a uracil.
yep.
So again, transcription needs 3 basic things in a reaction, of course. It needs the RNA polymerase. You need the free nucleotides, which of course are ribonucleotides, not DNTPs, but just RNTPs. And then you need ATP. And ATP, of course, is supplying the energy that is used up by creating those bonds. And then in translation, you actually need 4 ingredients in the reaction. And of course, you need the ribosomes, tRNA, which is carrying your amino acids individually, and then amino acids, and then ATP again. And so if you noticed in your binder, the student worksheet has them thinking through, well, what would have to be in these little tubes to be able to perform transcription and translation? And so they had to list that out and kind of really think through, oh yeah, ribosomes aren't used, wouldn't be used in transcription. So BioBits, I should let Allie talk about these, but she can, Allie, chime in wherever I make a mistake. But BioBits are cell-free, which means basically they've grown cells in batches and then they have stopped them in their growth and gotten them to extract just the useful parts, the parts that they want them to do. And there's lots of ways to do it. One, for example, is to engineer the bacteria to overexpress something so that it's making an abundance of whatever you want. And then you freeze and extract very carefully through an 18-hour procedure the extractions that you want, which then create these cell-free reactions. And there's so many amazing things that are now being used in cell-free reactions. For example, they can be used in biosensors because you don't have to have cold chain to ship things anywhere. And they're just literally for these, you know, they're this set of tubes. So those little yellow dots you see at the bottom of your PCR tubes are literally the BioBits. And so what do we need to add to our BioBits to do this lab? And that's, of course, we need to add our DNA, which you have in microcentrifuge tubes, and I have in these BioBit tubes. And then water, because we need to have a little bit of extra water in there to allow the molecules to move around. Allie, you want to jump in anywhere?
No, that was a really good summary of everything. Yeah, the extraction process is actually, it's not 18 hours. It's actually very, very quick. We just bust the cells open and take the innards. That's all we do.
So you're not trying to do like, because anyway, wow.
I mean, for more for more, I guess, involved applications, like if you're trying to make a diagnostics, if you're trying to do the biosensor, that kind of thing, we may need to do something fancier. But just for your students to be able to visualize transcription and translation, we just bust the cells open and just scoop out their guts.
Ooh, I like that. I may need that protocol. So let's talk about the gene sequence that is designed in our BioBits for today. And so if you look at this, we have a promoter, which basically turns on transcription. And then we have what's called an aptamer sequence, which is coded into the mRNA, and it folds in a certain format, which will then match up with a matching aptamer that's in the reaction tubes. We'll talk about that in a minute. After that, of course, you have the start codon and then the protein coding sequence. The protein coding sequence we're using for today is red fluorescent protein. And it's great because you can actually see red in the tube as well as visualize it glowing pink when you put it in the UV illuminator. And then you have the stop codon, which ends translation, and then you have a terminator. So if you look in the binder, materials have the kids, I thought this was excellent, Allie, by the way, if you wrote this, an excellent next page over, yeah. It has them think through, okay, what would happen if the promoter wasn't there?
It's on page 21, guys.
Thank you. What would happen, you know, if you had the coding sequence, but you didn't have, you know, the aptamer? So it makes them think and then be able to hopefully predict what they're going to see in their tubes. So how does an aptamer actually work? Oops, did I go backwards? Where did I go? Sorry. Okay, so when the mRNA is transcribed into DNA, the solution's going to turn green. And that's because the aptamer, and you can think of it sort of like, I think of it almost like a key and a keyhole, with the aptamer sequence being the keyhole, and then the green fluorescent protein has a little tag on it that matches with that aptamer. So that's the key that goes in, and then they form a structure. So only when that mRNA is present can the aptamer find its sequence and form the structure to then eventually glow green.
Just to clarify, there is no green fluorescent protein. It's a chemical called a fluorophore.
Okay, so it is a fluorophore rather than the GFP.
Yeah.
Great.
Yeah, so it fluoresces green. So when the fluorophore is free floating in solution, it doesn't fluoresce. It only can fluoresce when it's being bound up. And the only way it can be bound up is if the aptamer, I have slides that demonstrate this. I don't know if that would be helpful at all. But basically the aptamer is almost like a protein. mRNA can form secondary structures. And so the mRNA, so the aptamer sequence, basically the sequence is such that it will kind of fold up on itself to form a binding pocket, and that will catch the fluorophore in solution and emit the green fluorescence. So the only way you have green fluorescence is if you have the aptamer sequence in the mRNA form. The only way you would have that is if you had successful transcription.
That's great. So we're going to stop in a second and talk through this and answer questions, because this is, I'm flying high.
Yeah.
Just muted myself. So if we then think about it, so then that mRNA not only will bind to the aptamer and the fluorophore, but it obviously will find, ribosomes will come along and then translate that into a red fluorescent protein. But think about the timing. And this is why I think this lab is so great, is because it also gets into, if you're doing an upper level biotech class or an upper level AP class, AP bio class, you want to talk about, you know, RNA degrades more quickly than DNA. And so this lab also demonstrates that. So think about, you know, you first have to make mRNA, and that's going to then turn what? Green. And then eventually, once the mRNA is found by the ribosomes and is translated, once you get those amino acids and they're folded, then you'll get the red. And of course, there's a timing factor, which is great for the students to see that cells don't necessarily, they can't produce a protein in, well, they can, but they don't always produce proteins when we want them to. So I'm going to go to the next slide. And Allie, I think I have those slides for the broccoli formats. I think they're in the slide deck a little bit later. So
Hey, yeah, I have a very specific slide that I made. I don't think that's available in our curriculum that demonstrate the aptamer, because it can be kind of a tricky concept to wrap your head around. It's something that's used by scientists in their own research to be able to track RNA expression. So it's a very commonly used technique, and so it might not be something you guys have heard of. So I do have slides that kind of go, I just linked in the chat a webinar where I go over the entire lab, so you guys can check that out after the fact as well.
So if you want to think about your tubes, when you just have DNA, it's going to be clear. And then when you have mRNA, it will be green, and then you'll finally end up with a red fluorescent protein. So the first thing we're going to do now is stop before we do any pipetting or anything and answer questions. And I can't see everybody because I've got my slides up, so if somebody would help out. Yep.
I actually think it would work for people just to unmute themselves and ask a question. So no need to raise your hands or anything. And then the other way would be in the chat.
Great. Great.
Seriously, ask all the questions. We know what this was like the first time we saw it. It can be terrifying, especially when you feel like you have to explain it to someone else and you're still wrapping your head around it. So every question is a good question. We've asked all of them and we've messed everything up along the way. So.
And if you are shy, you always can ask a private question into the chat. So you just go over to that little arrow down where it probably says everyone, and you can find Janet's name on there and send it. And of course she or Brittany's, and send it to Janet's because they're in front of her, and she'll just answer the question and not call you out.
I promise.
You can always email us later too, if right now is overwhelming and you're sitting there looking at something later going, what? And keep in mind too, guys, we can provide you copies of any of this material and it's all available online. So feel free to mark it up. Like, this is yours to jot notes on, and like at this point I should say this, and oh, that was a great example, and oh, I really like how so-and-so explains this. Like, make this very user friendly for yourself later as you're going through it, so it feels like good teaching resources.
All right, so it doesn't look like there's a lot of questions right now.
Can you do like just, okay, this is how it plays out in my brain, and I'm sorry, but so you gave me a DNA sequence, right? And then to prove that it's transcribing, you added that whatever it is, aptamer to it. And so when it changes colors, I know that I have RNA. I can prove transcription.
Perfect.
Okay. And then after that, then to actually see the protein, I'm going to see green.
Yes.
Okay.
No, you'll see
No,
red.
Red.
The protein is a red fluorescent protein. So transcription is green and translation is red.
Transcription. So, okay. All right. So
So
the
that's
aptamer turns it green, and then the protein will be a red.
Yes.
Yes. So successful transcription will look green. Successful translation will look red.
And you can think about it as you tell the kids green means it's going and red means it's stopped.
Done. Good. Thank you.
You're welcome.
And that matches the slide pictures if you back up that one slide too.
And Sheena, that was a perfect question because that's what the kids always ask you. They're like, whoa, wait, what? What?
So if you guys are ready, I have my tubes and everything already. I'm going to kind of tip my camera and hold things up as I'm pipetting. So the first thing that you want to do is you want to get your strips like this. And you should all have individual strips of 4, and just make sure that they're the ones with the yellow in the bottom, not with the white. If you got the ones with the white beads, they're the wrong ones. You need the ones with the yellow beads.
So we should have because you've got all those evidence. Oh, yeah. Oops. So maybe you should check it. Okay, so there we go. So stop sharing. So does everybody have your tubes? Give me a thumbs up or shake your head or waggle
I
back.
see a lot of tubes up.
Yes.
Yeah,
Good.
great. So Clark County, I see somebody working furiously. You guys good?
So just a little viewing tip for you guys too. If you click on Janet's screen and go to the 3 white dots, you can pin her video and that will make her screen larger to you so you can see it better.
Because I need those for the directions.
And of course, you have the directions for this lab on page
Starts on
13.
13, I think.
On page 13. So the first thing that you want to do is you want to label your tubes, and you don't obviously, or maybe not obviously, you want to remind the kids not to label the tops of the tubes because it's like Petri dishes. It's too easy to put the tops on in a wrong direction, and then you have no idea which tube is which. And so I always label my tubes, and you can see them here. Let me see if I can pull that up for you. So 1, 2, 3, 4. And I've just gotten in the habit of labeling my tubes at this top sort of ring area, not down here, because when you run PCR reactions and the heat of the heat cycles and that condensing and going back and forth can often take the ink, even if you use Sharpie, take the ink off the sides of the tubes. And then you're ending up with tubes that are unlabeled.
You're labeling the empty tubes, right?
Yeah.
Or you?
Yeah. These actually have a reaction in them, but I labeled the empty tubes.
Okay,
1,
so
2.
we only need 4. All right, gotcha.
Yeah, your
So-
tubes should have the BioBits reactions in them. They're not empty.
Yeah, so they should have those yellow.
Got the yellow.
Yep, that's what you want.
Label them 1, 2, 3, 4. All right.
At the yellow, you label them 1, 2, 3, 4. Thank you, God, for that fan. Welcome. Oh, the kids are gone if you want to take your mask off.
They're gone?
Yeah. Yeah,
I
the kids.
swear we've been together like this whole time, you guys. We are practicing good social distancing, I swear.
But now you can see that I've labeled these bio beds. All right. So the next thing you want to do is sort of just kind of tap it to make sure your bio bits are at the bottom of your tube. And then you want to go ahead and prep and make sure you have the water, the two DNAs, DNA A and DNA B, and then the kanamycin. Okay, so now in tube 1, you're going to be creating your negative control. And of course, this is a great time to talk with the kids about why you need a negative control. Anybody have good ideas of what to do? And you're going to add, I can't see, what am I doing? What you need, baby. So to tube, yeah, there we go. So what is the purpose? What do we expect to see in tube 1, which would be what I would ask the kids for a negative control? Everybody's busy pipetting.
Before you actually pipette, there's a couple of best practices you probably want to tell your kids. Janet already mentioned you want to make sure the pellets are at the bottom of the tube. Sometimes they can get caught in the cap, so you want to tap your tubes at the table to make sure they're down there. And when you're adding the liquid, you're adding the reagents to the tubes, you want to make sure your pipette tip does not touch the BioBits pellets. If you jam your tip all the way down to the bottom of the tubes, the pellet might get stuck. So usually, if you look at the protocol, I think it might be in there. The best practice tip is basically to touch your tip to the wall, the side of the wall right above the pellet, and then let the liquid drip down. And when you're also opening your BioBits tubes, you want to open the caps very carefully, one at a time. If you just rip your caps open, the pellets might go flying. So just quick tips I've seen people have messed up before.
Like mine just did. Oh, back again. Okay, so if we've labeled our tubes, so let's talk about what will be going in these tubes. So tube 1 is our negative control. And so we would expect to see what colors with just water. Nothing. I just saw Laura go, nothing. That's great. And then tube 2, we're going to be putting in our DNA A.
Nothing.
Yeah, perfect. Can you flip to?
Hey, question real quick. How important is it to keep this on ice or all these tubes? I mean, how long can they sit out, or are they hard? Can you hear me?
Yep.
They can sit out for 3 weeks at room temperature, so you're fine.
Oh, okay. So we don't have to put them on ice. All right.
That's one of the great things about this lab is you don't need the cold chain storage. And so it's perfectly okay. I just have them in the racks.
Gotcha.
And DNA, as you know, is very stable. So in tube 2, okay, everything is there for transcription and translation to occur. And we're putting DNA in that has the full coding sequence. Promoter, aptamer that will attach to the fluorophore. Then we have the full start RFP protein, and then finally a transcriptional terminator. Excuse me, whatever. So then in tube 3 is really interesting, and where you can draw in for kids, if you were teaching a unit on how antibiotics work, this might be an interesting lab to pull in, because kanamycin, the activity of kanamycin, it disrupts the activity of the ribosomes. And so you're going to be adding kanamycin along with the DNA to tube 3. And of course, then what's going to happen is with the ribosomes interfered with, you're obviously then RFP will not be able to be made properly or folded properly. And so again, you should not get red fluorescent protein. You should only see And I'm going to assume everybody just said out loud green. And then lastly, in tube 4, this is your experimental tube. And so we don't know, it's a mystery. And so you'll be able to tell a little bit today. And then when we show our magic reaction tubes, you'll be able to see what it should look like tomorrow. And then I would encourage you to go ahead and check, even if you can later today or tomorrow, obviously, to see what the reactions look like. All right, so I'm gonna go ahead and start pipetting. If we go here. So this is where the kids, you can pause and have the kids actually stop and make their predictions. We've kind of talked through our predictions, but I think what is critical is for, especially as they're developing their CER, which is the lab report out that's included in this lab, and Dr. Cantrell is going to talk about that a lot, is to have the kids think through why and write down, not just say why, but actually write down why they think these things are occurring. So if it's green, why is it green? If it's red, specifically why? It will give them the basis then to develop their reasoning later on in the lab.
Something fun that we do, I mean, if you have laptop, like rock top tables, those chalk markers, or if you have, you know, the big kind of whiteboards that you can have cut down at Home Depot or Lowe's for way less money than what they charge you to buy them. Those are great places for kids to do that. They kind of write it out of the way and then have it to look at later so they can come back and look at those. All right, here was my initial claim. So I think sometimes the kids get hung up on the idea that their claim has to be 100% right. And our size is not about the right, it's about the thought process, which they can go and correct later if that thought process was wrong.
Okay, so my pipetter looks probably a little bit different than yours, but I've set it at 7 microliters. And then I have my reaction tubes. And so I'm going to be adding 7 microliters of the water to the first reaction tube. And, you know, last week where we pipetted up and down to mix, don't do that this time. So with these small reactions, if you introduce bubbles, it's going to interfere with the molecules finding each other to create the reactions. And so I just laid it, just like Allie had said, I laid it along the side. Let's see if I can do this. I just put my pipette tip in, and then I laid it just along the side and ejected, so that the water then just sort of drained down to the bottom. And then we're going to go ahead and add the, I'm going to keep my same pipette tip and just do my waters across. So I'm going to add my 2 microliters of water to tube 2. So again, I've set my pipetter to 2.
And at this point, guys, like, it's really helpful for your kids to be able to do, but if you have enough pipette tips, you can also just do 1 tube at a time if you're worried about them messing up.
So you can see, so I'm going to try to get this up to the camera here. So 2 microliters is really, really tiny. And when I expelled it, you can see it didn't go down. It's sort of hanging out on the side of the tube. All I need to do is sort of tap it lightly, and eventually that water, the reagents will flow to the bottom. When we put the top back on, the lids back on, I can show you a method to sort of flick it down so that it will all go down to the bottom. But you don't need to worry about that right now. We've got another.
I have a question.
Shoot.
Okay. So where are you sitting the yellow? What is it sitting so it doesn't fall around?
Okay, so I'm using a well plate, but another great thing that you can use is an empty tip box, because the tip boxes that you have, the tubes fit perfectly in those rows if you wanted to.
Okay.
Are they also using the P51s to visualize the reactions?
I have this. Did some of you?
I was gonna say if you had,
Meg,
because these make a nice tube rack too if they're using these, but if not,
Oh yeah.
Yeah, they don't have those.
okay. I mean, you could use the inserts as well, lie the tubes on their kind of tilt on the side if you want. But yeah, tip boxes work as
Of course, it is a good sterile technique to leave your tip box open, but in this case, we're just going to have to go with it. Okay, so then you're going to go ahead and instead of water into tube 3, you're going to be adding 2 microliters of kanamycin. And this is a great lab for the kids to also think about good sterile technique. That if they're opening microcentrifuge tubes, they shouldn't touch the top. They should be careful as they're going in. You should check, always check, especially when you're doing these small amounts. Look at the tip and make sure you're actually getting something in the tip. Some micropipetters don't work as well as others. And I found when kids, especially honors bio, no hurt intended, have used my pipetters, that they don't often work with 2 microliters really well. And so you want to make sure the kids are checking that they're actually getting the reagents that they need.
Okay, I have one more question.
Sure.
I'm one of those that I don't care if you call me out.
No, you're good.
Okay, so in my yellow tubes, in tube 1, I
Yep.
put my water,
Yep.
and that was 7, the 7 milliliters that I put here.
Yep. Perfect.
So, and then in tube 2, in this, where do I use my distilled water, or I don't know if I really have anything left in here.
You should have a tube of water.
Yeah, I have that, but I used the 7.
Oh, there isn't any more in there?
Well, it looks like maybe it's awful small if it is.
Then I would go ahead and just use the distilled water. That's okay.
You only need like 2 microliters, which is a
Yeah.
tiny amount. That probably is enough.
Okay, and then so I haven't used the DNA A and DNA B yet, correct?
Yep.
Okay. I'm gonna close it back up.
So that's a really common question, is this tube doesn't have anything in it. But you have to realize that sometimes these really, really small amounts don't look like anything, but they actually are the reaction amounts.
Something came up whenever I did
Oh,
it.
perfect.
Perfect. It's
Janet, was it a purple tube for the water that Kian put together?
He did not leave me a code as to what went into what color. I love Kian. He did my lab aide did all of the pipetting for all of this and was so organized, but I did not get a code for what colors went with what reaction tubes.
But it should be labeled.
But it should be absolutely labeled.
It said nuclease-free water, I believe.
Yes.
He wrote all of that on there.
Oh, did he really? Okay.
I think so. Or like NUC-free water, maybe. I see water. All right, Sheena's holding up water.
Okay. So are we ready to pipette the DNA yet? I
See
got
one
Keith that said yes.
wiggly thumb.
Do a yay. Are we? I got Brittany.
2, 3.
Are we supposed to observe the tubes in a blue light?
Not yet. You need to put the DNA in first. Well, oh, wait, sorry. Yes. Okay, stop now. Thank you. So you can take these reactions, because the kids might think, well, they're going to glow green. You're doing some kind of magic voodoo thing. You can absolutely take your tubes and put them in the illuminator. And I'll do that and try to show you. Let's see, without making it to your stomach. Okay, so I'm using, let's see. See not that. That's does that work for you guys?
Ah, look at there.
So to really visualize the colors, that there weren't colors there, you need to turn all the lights off.
Janet, can you show how you put that thing in again, please?
Oh, sure. The tubes in this.
The orange thing into this thing.
Oh, perfect. So I think this is also, oops, I sound like a salesperson for Mini PCR. I promise I don't get a kickback. So you take your blue gel and then your orange rack. And I was stupid earlier today and I was like, it doesn't fit. And then I realized, dumb. So turn it sideways. And then to use it, you just push the little light button. Oops. Cynthia's got it. What? Oh, I don't know.
I just want to say we hear from industry all the time, and that it is so important for employees to be able to be safe. It's the number one thing on the job. And so you'll see when we go over later in our essential skills and competencies that aseptic technique is on there. So I'm super proud of you guys. I see lots of gloves being used, and you know, we're okay with what we're using right now, but goggles are always a great idea too. So
Yes,
that's some of the stuff.
Meanwhile, we've got no goggles or gloves on.
I mean, I'll just point out, those aren't UV transluminators. Those are safe blue light. You're not dealing with anything
Safe blue.
UV, so it's all safe.
Yes, sorry. Yes,
Right.
thank you.
So-
Yes, I'm having a glove shortage. So I'm not using gloves because literally I'm counting every pair for my biotech and iGEM kids.
Megan can get you some more if you need some.
Yeah,
We had
if you
a
don't mind
really nice
latex.
donation.
That would be awesome.
I'm not
Yeah.
seeing a color here, guys.
Yeah, I can barely see something underneath tube number 3, and I even crawled underneath the desk where it's really, really dark, and I'm not
You're
colorblind.
not supposed to see anything yet. You guys
Yeah.
just added the reagents.
No, it's been a while since I added mine.
Should it be?
Did you
How long
heat
should
it?
it be?
So, okay, because it says that you should be able to view something, and then like, how long are you wanting us to heat it for 37 degrees Celsius?
So you can use body heat. You don't have to use an incubator. It's usually you can usually see results in like 15 minutes. The longer you keep it though, the brighter it gets.
Oh, cool.
So I'll show you so you can look, see if I can pull it.
Cool. All right.
In the PowerPoint later, I showed, so this is, well, this is what I took this morning when I ran my magic tubes, the ones that are I'll show you later. And you can see that basically the fluorescence or anything that you're seeing is really the reflection off the tube. It's not true fluorescence. And so I wanted to see how long it took. So I took pictures every 5 minutes to record. And it really was right about that 15-minute mark. At 10, I kind of saw some green developing, but then by 15, I got a full develop. And I put them inside my shirt to incubate them. Kids could hold them in their hands. Don't laugh at me. I'm just like,
Sorry, I don't have a bra, Janet. I'm useless.
Come on, Steve.
I'm not putting it down my trousers. All right.
Please don't. Please don't.
Oh yeah, I see a color now. This is great. Nice.
Protocol. Yeah. Okay, so has everybody started their reactions? Are we ready to go on? I see head shaking, thumbs up. So I'm going to give this to Brittany to hold since she didn't have the pleasure of incubating the BioBits this morning. And then while they're incubating, I'm going to share screen. Nice and warm in there.
What you looking for?
Oh, because I'm not on that. Like, I have a word.
Wayne County said they're struggling a little bit.
What do they need help with?
They said just hold on, please,
Oh, absolutely.
so we can take a little chill break.
Yeah.
This would be a good time to like, do people have questions? What kind of questions can we answer for you?
I have a question with this thing. So this is just it?
No, you take the rectangle, this,
Yeah.
that comes off,
Okay.
and you should have gotten a little wedge shaped thing that has holes in it.
Let me go check.
Yeah, it was either in the bag with your blue gel or it was in the box with the blue gel.
Oh, it's in the box. Okay, sorry. I was
Yeah.
like, I don't know what they're talking about. I can't make this box a triangle. Okay.
Long orange box thing, yeah.
Okay.
The triangle makes it easier, but in a pinch, you can also just use the box as is.
Okay.
Yeah, just set it on top.
And then now, I just sit it on there.
Yep.
Take the clear box out.
I'm not good at puzzles.
Yeah,
Yeah,
take
take
the
the
gel
clear
spray
one out.
out. And so you just have this.
Yeah,
Yes.
There,
yay!
there you go.
Just making sure we haven't done anything with the DNA yet, right?
Oh, no, please add your DNA.
Okay. What page is that on?
So you're going to add 5 microliters of, it's on page 13 and 14. 13 and 14. So you're going to add 5 microliters of DNA A.
Hold on. Randy, can you see this? Is it too far away? Here, I can pull up.
Oops. Wait, you're tipping. Don't tip it. There you go. Randy, does that help?
Yeah, I'm just figuring out how to make the screen bigger. I don't know how to do that.
We were supposed to be doing this in person.
Y'all,
All right.
so sorry.
We were supposed to start this with a wine and cheese party.
2020 is not fun, y'all. Yeah, we planned all of this to be in person and it was going to be, all the hands on.
Yeah.
I think everybody's doing awesome, is awesome. This is tough, and especially after a full day of work. So thank you to our instructors and thank you to our participants.
No, thank you, participants.
Yeah.
Are you trying, Brandy, trying to make Janet's screen bigger? Is that what you're trying to do?
I found
I'm
it. on
typing the amounts into the chat too.
Thank you,
Allie.
Allie.
Yeah, I see on the bottom of page 13. Tube 2 gets 5, tube 3 gets 5 of DNA A, and then tube 4 is 5 of DNA B.
Yes,
I'm working on that now.
great.
Sorry.
No, you're
No,
fine.
you're good. I'll just take the moment to air condition myself.
It's balmy in here.
We're on COVID quarantine right now, ending our 2-week quarantine on Wednesday. The kids come back, but I swear the county office, to punish us, has turned the air conditioning back because there aren't kids in the building.
Yeah, there's not as much body heat. So they assume you don't need it at all, right?
That's right. That's right.
Are you guys doing in person and traditional at the same time, or are you all or none?
We're everything.
Yeah.
Where kids could choose to do all virtual, they could choose to do all in person, or they could choose at the high school level a hybrid, so that they could come in for some periods and then be virtual for other periods.
Oh, wow.
It's really fun, guys. It's not at all hard to keep up with. And then all the kids on COVID quarantine that are face-to-face are virtual, but not the same kind of virtual as the kids who are actually virtual.
because the all virtual kids are on county pacing, where the kids that are just quarantined from your classes are on your pacing,
Which is not the same pacing.
which is not the same pacing, because why would that be?
Yeah, our county just, it's, you can be virtual, but you're still following the traditional curriculum of the same student. So all these labs that I'm doing with the kids, like if I can't find a simulation, then they write me a nice paper about whatever we're doing. 'Cause
Oh, that's rough.
I can't monitor. I tried sharing, like, why don't you watch us do the lab? And that was a lot of accidents spilled. I lost 2 computers that way.
Oh, oh,
Yeah.
So they just make me pretty posters and write me papers.
aw. My friends that still teach in Pennsylvania are having to teach to the virtual kids and their in-school kids at the same time.
So that's what I'm doing. It's awful.
I don't know how God bless you. All
Like
love-
I have to turn my computer around so that the kids can see me at the smart board. And then I have like 3 computers going at one time to show different things. And then my favorite part is I say, "Hey, can you guys hear me?" And my kids in front of me are like, "Yeah, we can hear you, Miss Adam." I'm like, "Yeah, I know you guys can, but I'm talking to my virtual babies." Or can you see what I'm writing, guys? And they're like, "Yeah, Miss Odom." And I'm like, "No, I'm not talking to y'all. I'm talking to these guys." It's fun.
And you're here with a smile on your face. So
Yes.
you really are amazing.
Yes. Okay, so does everybody have your reactions now cooking? Awesome.
Thumbs ups from everybody.
Yeah.
That we, especially ones we can't hear from. Clark County folks, y'all okay?
I see a wave
Okay.
Yay.
getting there.
Good.
So I'm going to go ahead and share my screen for, oops, where'd it go? Nope, there it is. For the next slide. So obviously, you can incubate these tubes any number of ways. I let mine, like I said, I held mine not in my bra. I just put it up on my shoulder, smart Alec. And for 15 minutes, and just took it out every 5 minutes to take a picture because I wanted to see what the reaction rate was like. And then I continued like every hour to take a picture to see when, over the next few hours, to see when I could see the actual changes occurring. And so you can see down here at the bottom, right? That's what my tubes looked like after the initial 15 minutes. And so that's that green fluorophore is awesome.
It's not just a little green, like you can really tell, and the kids can really tell too.
I can also probably make that bigger because I realize now looking at a computer screen, that's really tiny. There you go. Nope. I deleted them because they were all the same. I have like, I should have done that. I'll do that next time. Oops. Megan texted us. Yeah, I'm responding. So then at this point, I got to watch our timing. We are at 5:15. How are we doing on our timing?
Not great, but that's
Okay.
all right.
Okay. So then at this point,
We get what we get.
you would then wait, like I said, anywhere between 8 and 72 hours, and ta-da, magic occurs. And so we're going to pretend it's 24 hours later. And this is what my tubes, again, looked like. Yeah, really cool. And what was, like I said, what was interesting, about 4 hours in, what I saw occurring in tube number 2 was it was really in between colors. And then finally, eventually and I didn't have to incubate it. I just left it on the lab table all day.
What am I doing? Can we show them the real stuff?
Oh, yeah. So,
Oh. I'm gonna show you the tubes, guys. The real deal. Now I might just be able to do it over here on mine, so you don't
Oh,
have
yeah.
to stop sharing your screen.
oh, it's flipped backwards. I have the tubes in the wrong order. You're like, Janet.
No. There we go.
You look like ghosts.
I know. I'm trying to get my laptop close enough to me, but can you guys see that enough to tell what's happening?
Try, oh, where are you? Here, I'm gonna stop sharing my screen.
Oh yeah, y'all find Brittany and pin her for a minute.
Yeah, I'm
I think
trying.
you might be able to see it pretty, I think so. Can you guys see pretty well if I'm holding it like this?
Yeah.
Okay, so now you can see what's going on in tube 4,
And
right?
you can also take a screenshot right now of your computer so that you have a what my results should look like kind of deal. If you like to have that like me.
I got it if anybody needs it. So I'll put that in the drive.
Okay, and I was gonna say, I'll send Megan my pictures.
Perfect.
But Megan, I deleted a bunch of them because they kind of looked all the same.
Yeah, fuzzy.
Next time, when I do it with kids, I will definitely do a time lapse. I think that would be really good.
Sweet.
Okay, all right. I think we're ready for lights. Yeah. You guys okay if we put this down? I don't do well without verbal feedback. There you go.
Off. All right, so in my schedule, I had kind of thought this was a great stopping place for a break in case people needed coffee or restroom, and then we'll jump back in with a few more introductions and then talk about the lab reporting of this and the aspects of the problem solving and the thinking that goes along with this lab.
And we'll talk through some resources too that you'll then have shared with you after. We wanted you to see them first before we just slammed you with a bunch of stuff.
So break time, dance time.
I do have a question.
Sure.
So while mine's sitting
Yeah.
in here, my light's on, do I need to put it in my shirt as like, what do I need to do to incubate it? Or is it incubating on its own?
So you can incubate it inside your shirt or hold it.
Make
It
go
just
faster.
makes it go a little bit faster, or you can leave it on the lab table.
Okay.
For the first 15 minutes, so you do want to have it heated because the aptamer is optimized for 37 degrees Celsius. If you do try to incubate it at room temperature, it won't fold as well and you won't be able to see the green as well.
Okay.
Yeah, and then to visualize, obviously you're gonna need to make the light, make the dark light.
I mean, but once you pass that first 15 minutes, then it can be at room temperature. It's fine.
Okay. Thank you.
I have a question for you, Allie. How do you see, could students potentially use BioBits for independent research projects beyond just this particular lab? And like, how do you see them doing it?
Yeah, when I was developing this in my PhD, I actually published 3 papers, different prototype. I can link them in the chat after I'm done talking. But yeah, there are lots that you can do with BioBits besides the central dogma. We're working on developing new BioBits labs now. The next lab that's going to be released will be our protein structure function lab. That's going to come out later in the year. And we have lots of ideas for other ones. And definitely we've had students and classrooms contact us about using this as like independent research, because theoretically you could just add any DNA encoding for any protein and it should be able to make that protein. Obviously there are limitations and restrictions on that, just how well it's optimized. But yeah, it's definitely something that students could use for independent research if they're interested in. We only sell it as kits right now. So if you wanted to get your hands on BioBits, you would have to purchase the Central Dogma kit and just use the pellets. But the DNA could be used as, like, say, a positive control, making sure that your proteins are working. Yeah, I'll pop my email in the chat. If you have any questions on this, you can contact me, let me know, because there are certain proteins you can't make in this system. It's a prokaryotic based system. So for example, if you try to make eukaryotic based proteins, that might be a little tricky. So let me find the papers, and just keep in mind these papers I'm about to link. These are all prototypes. They do not exist as a actual product yet, but they might give students ideas of the different things that the system can do. And these papers are all open source, so they're not behind a paywall or anything.
I'm going to brag on Allie for a minute. She works in one of the premier labs that's on the very forefront of the cell-free technology. She won't tell you that, but her lab is doing amazing work in this frontier. So I'm just so privileged to have her on.
Thank you.
And I watched one of your webinars. Mini PCR does great webinars, and I've watched any number of them. And that's how I learned about the Central Dogma, was I watched Allie's webinar.
And guys, if your kids have some prep, they're appropriate for them too.
Yeah.
Like they're definitely very student friendly and they do a really good job explaining. So feel like that's something that you can, we've got all this linked in resources.
Yeah. I just linked it in the chat, and yeah, they're geared for students. We actually make worksheets that students can use to follow along with the webinar. And yeah, a lot of them are live demos. So if your students can't actually do any of our labs, they can at least watch and follow along. Our results are super visual, so they can at least get data and analyze and that kind of thing.
Are we incubated yet?
It's been 15 minutes.
I don't know. I didn't take time. That wasn't my job. That was your job. Okay.
It won't hurt. We'll leave it 5 more.
Okay. So do you want to take over and do your CER?
I can. Yeah, this would
Okay.
be a good time to do it. Let me just use your computer. It'll be easier.
Yeah, I was going to say, we're going to shift seats.
Okay. All right, guys. So, like I said, we have some resources I'm going to show you right now. And then either Megan or Kristen is going to share them with you. I don't think you guys have rights to these yet. Give me just a second. And I want to share. Is this that screen, baby, where I can get to? Okay.
Yeah. Oh, and while you're doing that, I have in my scribbly way all of the handouts, all the mini PCR handouts that came in your binder. I have written out potential answers, and I'm happy. My scribbles are scribbles, but I'm more than happy to share those so that you can see some sample answers, or if you have any questions. I actually had to look something up a couple of times.
Yeah. So, guys, can you see? I think we're okay. So some of you may be familiar with claim evidence reasoning, but we in secondary schools right now are really trying to push for more scientific literacy and more writing in the sciences. And so being able to fully articulate when you're explaining a concept and when you're saying, okay, well, we did this in the lab and this is what we got. All right, what does it really mean? So what we have found and what a lot of research suggests is a lot of times when kids are doing labs, there's a concept and they're doing this lab and they're not connecting how the lab and the concept go together. And so what we're asking them to do is explain what are, these observations that you're making, this data that you're gathering, what does it mean? So essentially making sense of the scientific process. And so we put this, there's argument-driven inquiry, which is where this comes from. There's tons of resources out there. I've got lots and lots of them in a folder for you guys, sample rubrics, sample graphic organizers, different ways that you can introduce this, but I wanted to go through just kind of basic CER, which is claim, evidence, and reasoning. And so what you do is you pose a question to your students. Why is this happening, or what's going to happen? In this case, we could say, what are we expecting to happen in our different tubes, or what is the role of something in? And so their claim is going to be, a sentence that answers whatever essential question you've asked the students. And then the evidence is going to be the data that they gather in the lab. So often they're gathering data, they're putting it into a pretty chart, but they're not really necessarily having to make much meaning from it. And so the reasoning piece asks them to pull a scientific principle explaining why their evidence supports their claim. Now, what we also tell students is you don't necessarily have to be right, but sometimes the gathering of the evidence and doesn't support your claim, and that's okay too. But it's pulling the whole process together so that they're having to tell you, all right, here's the guiding question for your lab. What are you claiming to be true? How do you know it's true? What does that actually mean in the world of science? How do these things connect together? So a really good way to explain this, and I always like to start with my kids. So usually I'm introducing this in a 9th grade biology class. And then I'm expecting tiered levels of it as we go through honors biology and AP biology and biotechnology and that sort of thing. But I like to introduce with something that's not even at all scientific. So just something really kind of simple and easy. There's this great little video. It's My Dad's an Alien. So it's very, very short. So I'm going to show it to you.
I have evidence that proves my dad's a space alien. He speaks a weird language. He drinks green stuff. He says he's from Albuquerque. I'm not buying it. I mean, just look at him. And one more thing, he has a spaceship. The evidence doesn't lie. My dad's an alien.
The highly advanced Audi A6, named to car and drive.
So super short and sweet. But then I asked the kids, all right, so her claim is that her dad's an alien. Well, what evidence does she provide and what is her reasoning? And so that kind of talks them through the process of claim, evidence, reasoning in a way that's not scary. So that then when you're working them towards okay, now you're doing this lab, I've got an essential question on the board and I've given you a set of instructions, a protocol, go, because our goal is to get them comfortable with doing a lab and working through it on their own and being okay with having to read and reread and work together as a group, and also okay with being wrong. So I don't know about your students, but mine super struggle with the idea of what if my answer isn't right? Okay, buddy, welcome to science. So we really like to get them kind of thinking about science in a non-threatening way so that we can then release them into the whole world of claim, evidence, reasoning. And we scaffold in the beginning and really work through together. Okay, guys, what's our claim? And I will go through groups, and I'll have lab aides go through groups, and we'll have the different labs talk to us. Okay, what was your claim? All right, now we're gathering evidence. They have to talk to me about the evidence, and then they have to maybe do a sign off where as a group they have to explain their reasoning. And so you have like a checklist that you go through with each of your groups as they learn to step themselves through this process. But ultimately there's great stuff from ADI, all kinds of different templates, things that you can even turn into posters to put up in your room or to hang on a bulletin board to refer them back. But what I love about this is you're providing a guiding question and you're letting them think through and work through the process of science. We all know we learn science by doing science. And so often I think kids are not given enough opportunity to do and fail in a supportive way because their lab skills aren't great in the beginning. And I mean, geez, by the time they get to biotechnology, you're doing all this crazy complicated stuff. You aren't always going to get perfect results. But the idea is that you can think through and claim through and provide evidence for and have to think about science and think about what it means. So, lots of different samples, guys, but, guiding questions can be, all sorts of different things. It can be wet lab, it can be dry lab, but you're giving them the question, they're making a claim, and then they have to figure out what kind of data do I need to gather and what do I need to show you as evidence so that I can reason out this process. So lots of different samples of ways that you can do this. What I wanted to show you guys were some whiteboard samples, because I love the whiteboards. The kids love the whiteboards. Home Depot and Lowe's, usually you have probably a business partnership with them. Check with your bookkeeper. So they will take, if you try to buy whiteboards from an educational supply company, they're insanely expensive, but you can just literally have Home Depot or Lowe's cut big sheet boards. It's like the backer boards that they put in prefab bathrooms, showers, and you can have them cut them down for you. Are they quite as nice as the other ones? No, but they're also like significantly less expensive. And so when I was the department chair here, I had everybody that wanted a class set of these for their labs. Everybody's got a set that wanted a set. So most all of the classrooms have between 16 and 20 of these in their room. And so they will go through, and it might be that even, you have a lab that's over a couple of days. And so they've got this nice big board that they're working with as they're working through their claim and their evidence and their reasoning. And so a lot of times too, I don't know about you guys, but taking up 150 lab notebooks at one time and trying to grade that, it like my soul dies a little, and it's important, and that they need to be able to think through and articulate. But I've had great results with going around to different lab groups and having them walk me through their CER process, and making sure that I'm asking lots of different kids in the group to do that too. So I know that it isn't just Janet who knows all the answers, but that, Kristen and Megan can answer questions as well, but it gives everybody an opportunity to discuss and think through the process. They also love, like I was saying, chalk markers. Those are kind of expensive. I splurge on those every once in a while because they love them. But I'll have them do like a CER on their lab note, like on the tabletop, and that serves as their lab write-up for their notebook. And so maybe they'll take a picture and have to drop it into a Google slide or something like that. But it's just different ways of reporting out. I've also, in the folder I'm going to share with you guys, put some very traditional ways of reporting out. So, traditional like components of a lab and what you would like want to put in there. Again, I know some of you guys have lots of experience with this, some might be a little less so. So there's all sorts of materials in the folder. Please feel free to use, modify, or delete. I just wanted you guys to have lots of different resources. So again, this one looks a little bit different, claim evidence reasoning. What I like to do with my kids, if I'm having them write out a CER, what I will have them do is color code their claim, their evidence, and their reasoning, so that when they're writing out a big report, I can go in and they've got a key and I'm like, all right, I know the claim is in green and the evidence is in yellow and the reasoning is in pink. And so I look for those sections, and it helps me get through the process a lot faster. But lots of different samples. Again, here's a great example of a whiteboard. What you can also do with CER is you can have them explain to each other, okay, group 3, we're going to talk to the class. What's your claim? What's your evidence? What's your reasoning? And so what I will do with that is either like write group numbers down or just do a random number generator. And that's the order in which I call on groups, because obviously it's harder to go first than it is to go fifth, because a lot of times their claims are very similar and they're, taking notes on like, oh, they did a really good job explaining it that way. I want to think about things like that too. I've got some links to what I really like is when you get your kids thinking and you get them to a point, you can really use the CER process and take it to a next level of adding almost an essay component to it. And so, let's see, can you guys see that? Maybe a little. So I've linked in a slide player here for you as well that talks them through, and there we go, through the essay component of this. So how is it that you would turn this whole thing into like more than just a one sentence and a table and a, okay, this applies to this scientific principle, but really thinking through and writing about what they're doing. And then I've linked some, hyperlinked some more resources here for you as well. So a student graphic organizer, maybe the first time you introduce claim evidence reasoning, and checklists for students to check themselves. So, hey, did I do this? Did I do this? Did I do this? Does my claim answer the question? Does it, is it more than just yes or no? Is it in a complete sentence? Can I gather data to actually answer the question with the claim that I've made? So I have found all these resources to be really helpful. What I want to show you guys is It's not this thing that's pinned right here. Hold on a second. But if you go into, you're going to get a folder and it's called management and resources. And I lost my mind and put like a million things in there. So again, feel free to use, modify, or delete, but you've got all sorts of things that you can have your kids use in their lab notebooks. Really great Cornell notes to get kids. I like to get them thinking with regards to, okay, we're taking some notes now. You need to go back and look at your own notes and answer, like write some questions down that you still have about information that we've covered. And, just setups for lab notebooks, pre-lab templates. I don't know about you guys, but I've always struggled with my babies when they're first starting out, getting them to really write and think about, okay, so I'm going into this lab. I need to know what I'm doing. A pre-lab is more than just, okay, I pretended like I read it before class and now I don't know how to do anything. And it's Dr. Cantrell, Dr. Cantrell, Dr. Cantrell. I'm like, there has to be some level of like preparing them and housebreaking them to be in a lab, or they, don't know what they're doing and it's not as fun as it could be. Okay, what sorts of things do you guys have questions about? I know we're going through stuff, we're flying through stuff super fast, but I'm just trying to make sure that we have a chance to cover everything and we don't run out of time. Again, this is not, our hopes in the future is that we're gonna be able to continue to offer things like this and that maybe one day we'll be back to normal face to face. And I appreciate you guys being so generous with us as we are done lots of teacher workshops, but never one fully virtually.
So I was thinking it would be helpful to go ahead and walk through a claim evidence reasoning for this lab. So Brittany, I bet you're incubated. So shout out somebody out there. I put it in the chat, but maybe you want to type it in the chat, or maybe you just want to share it. Just yell it out. Is what statement would you make if you got these results? Whoa, that almost was deadly. Trying to walk over a dog.
Yes, I have a rescue in here. I think we told you last week, who's a little bit attached. So has to stand underneath us while.
So let me turn off the light.
Let's see that.
So there are our tubes. And that's of course just after the 15 minutes. And then I'm just gonna exchange out the tubes.
So let's pretend like you've got kids, they've set this all up, they've made some initial predictions, and this is, 24 hours later when you're having them walk through a claim evidence reasoning.
And you can, sorry. Like I said, I'm dyslexic. So my number 1 is over here. And then my number 4 is over here.
You got it?
Yep, I got it. Oop, maybe I don't have it. Like Abbott and Costello, but in lab. Not as funny. All right, let's try that. So obviously, if we're, never mind.
There we go.
Obviously, if we're looking at tube 4, the kids are going to say, well, it's green. So what does that mean? So what is their claim? So there's lots of different claims that you could have for this. Why is it green? There's any number of things that could have caused that. So shout out, somebody give us a reason why tube number 4 is green.
Transcription occurred because of that aptamer reaction.
Yes, because of the aptamer.
Yep.
Or what's another claim that you could make about that?
Translation did not occur.
Yay!
Exactly. So that's what we also love about CER, guys, is there's so many ways to be right. And I think that's a really important lesson in science too, is that there are lots of different ways to explain the same thing and lots of different ways to be right and wrong. If you're looking at your lab notebook or you're looking at the handouts that you have on page 17, there's a really good predictions.
Yeah.
And this would be kind of what I would have the kids, I would have them set this up And then I would have them go through this kind of as a day one, and then come back on the second day and table 2, make their observations, talk about their processes, and then go through their CER. And so you can decide, what do you want their essential questions to be? You could ask them, why is tube 2 green? Or why is tube 3, I can't remember, 3's red, right? 3's red. 3's red. Or why is 4, if you want to pose it from a standpoint of, using the negative control, and then you've got the positive controls, positive control for transcription, positive control for translation. All right, now what does that mean about tube 4, since we don't know what that is, and we just have, the data to talk about it.
And so let's go ahead and take Laura or Sheena. Do we want to do the translation did not occur, or do we want to do the positive transcription occurred? So I'm just going to make a decision because we're getting close on time. So transcription occurred. We know, so that's our claim. So what would be the evidence? And this, of course, could be the table of the observations, but it also could be a description. And then the reasoning is where you get to see their thinking. And so I'd love to hear some of you. I know what I kind of thought that they might have done to the tube to cause it to just be green. But I'd love to hear some of your ideas for what's occurring in tube 4 that's different than tube 2, the red one.
Well, the evidence is it turned green.
Yes, the evidence is it turned green. And then you would also, for my kids, I would also require them to say the negative control was clear, the positive control was red, right? I would force them to walk through all of that. And then for now that we're getting to the reasoning, so their claim is that, our claim is that transcription did not occur, or transcription occurred, sorry, but translation didn't. So what would be some of the causes that could cause translation not to occur? Well, one that they're going to jump to immediately is, oh, they added kanamycin, or they added an antibiotic that interfered with the ribosomes and created it not to fold or not be created. But what are some other things, and your kids will be really creative with this, as to why number 4 could be green? So again, what do we know? So go back to some of the ingredients that are in the tube. So in your tubes, right, for translation to occur, you need what? You need ATP, which had to be present for what? Transcription to occur, because transcription also uses ATP. What else do you need for translation? Ribosomes. Well, they could have added something to make the ribosomes not possible. They could have somehow extracted the ribosomes out, although I don't think that's possible. I don't know. What are the things that they could have not put what? What gene could they have done? They could have changed the gene that was the right? They could have just put a blank DNA in there that was mRNA and something else, not RFP. And so by walking through this, they're thinking all the possibilities, or you're thinking all the possibilities, then they're explaining, well, it's green because. And so what do you guys think about this?
I like that it's a quick lab. Like it's not going to take you forever to go through all of those reasoning motions with them. Even if you split it in the 2 days is great, and I don't feel rushed.
Yeah. Well, and Sheena, what you can even, and I don't know how you normally assess or take up lab reports, but I mean, I would even have one person in the group, everybody writes their name on this. I do a Dropbox, one person takes a picture, one person drops it. And so if your labs look anything like ours, you have between 4 and 5 kids in every lab group and 7 or 8 lab groups in every class. Well, you've cut your number down to, 30 something, 40 labs instead of 160 something labs. So I think it feels very doable. And for me, balancing and making sure that they have a lot of lab experience and that they get appropriate feedback on it is really important, but it can be daunting.
It's deadly.
Yes.
I'll just say it
Yes.
for her. It's deadly. So Jackie said in the chat, it allows you the ability to think and get students to be creative while active in research. Absolutely.
Yes.
Because they're going to come up with some of the most bizarre answers to why it's green, which I think is awesome.
And things you wouldn't even necessarily think of. Sometimes you're like, oh, maybe that's.
Leads to divergent thinking. Yes.
Yes.
And alternative answers. Yes. Showing that science doesn't always lead you to the one right answer.
Yeah.
That there's lots of ways.
Speaking of, we're going to also share a folder with you guys on Central Dogma, and there's a bunch of stuff in there, and one of which is a template where you can either create sets for your class or have them cut it up, but it's all the key terminology around transcription and translation, and I have them do a big concept map putting it all together, and it's really amazing what those concept maps look like. They might be right, but totally different from one another, and I love that you can see the way their little brains work like that.
So we've run over on time, and poor Kristen is gonna be
by like
forced
7 minutes.
Be forced to do sum up workforce. But
I only need 5.
so please reach out.
Yes.
I'll answer any questions, do anything that I can. I'm looking forward to, I haven't actually done this lab with students, but I'm looking forward to doing it in a few weeks with my students.
So it was really, this discussion was perfect timing to lead into this. So the Bioed Institute, we exist to create bridges between potential workforce and the life sciences industry. And so the job that you guys have is to prepare your students for life and for careers. So in particular, we're focusing on life sciences careers today. So we are hoping to answer the question, what's the point in doing all this, basically? And so we have some research has been done, and the research is usually redone every 2 years. So 2020, as we know, you can probably guess it did not happen. So this research is from 2018, but still relevant. So just looking at the hard and the soft skills that are needed by the life sciences industry. And when we talk life sciences industry, that can mean pharmaceuticals, it can mean R&D, it can mean lab work, it can be medical science liaisons, so writers and communicators, public health, medical device. It's really the full gamut. So in breaking down these skills, we've got our top 10 hard skills and top 10 soft skills. So I will just preface this by saying every time Megan and I speak to somebody from industry and we want to know what skills are needed, they say soft skills. That is number one. So that's where the labs come in to play, because you address so many, if not all, of these soft skills when you're doing hands-on learning and especially laboratory learning. So I'm just looking over at my other screen here because I'm over 40 and you know how your eyes just go downhill. So I have to have it on my big screen. So I apologize for not looking at you. So our soft skills, communication skills, research and writing, the CERs address that right there. Any type of hands-on and any type of labs, you are going to address these soft skills as well as your hard skills. So quality and assurance and control, the discussion that we just had with the CERs, you're addressing why was that 4th vial the way that it was? And so that's kind of talking about the QA, and the QA is, there's so many positions right here in Georgia. Every lab, every R&D facility, every manufacturing facility has quality assurance folks. Organizational skills, I'm just reading these out loud in case you cannot see, but these are linked into your folder. Teamwork and collaboration. So even with Brittany's example of them writing one lab report, which I think is brilliant in terms of not having to take home so many lab notebooks, or even if it's a one-sheeter, is fantastic. But there's your teamwork in addition to communication and organization. Problem solving, being detail-oriented, troubleshooting, again, the CER addresses that, and then multitasking. And the labs do that as well, because there's always some piece that you're doing, then you have to wait, so you take care of something else. So when you're kind of defending or supporting what you're doing in the classroom and the importance of having hands-on learning and having labs in your class, here's your proof right here. So we want to provide this to you guys in case you need it to show your students, to show your parents or your community, and also to show your administration. Because we also know hands-on learning is, more expensive, but we are here to help mitigate that a little bit. Okay, so hard skills. And so again, these are addressing all of the life sciences. We've got laboratory research, quality assurance and control, manufacturing processes. And just so you know, Georgia is one of the larger manufacturers in the life science areas, especially with biopharma. So Takeda in Newton County, and then Recro Pharma in Gainesville Hall County, and then Dendreon in it's Henry County, are some of the larger areas, and they need all levels of workers at all education levels. Okay, chemical analysis, validation, cellular biology, research methodology, drug development, clinical research, and project management. The Google Suite has some really nice project management software if you're ever wanting to delve in a little bit deeper, and it's also a really nice way to collaborate with your English counterparts. Can you see this next document? I never know how Zoom works. Okay, so the essential skills and competencies are something that are specific to Georgia BioEd that Megan and I developed last year in just combing through as much as we could on Indeed, and specifically in evaluating bench work types of not bench work, technician types of positions. And so these technician positions were anywhere from high school graduation equivalency up to doctorate level education. And the top skills that kept on, there were just certain skills that kept on recurring. And so that's when kind of the light bulb went off, and we're like, is this just this essential set? And so that's where we came up with essential skills and competencies. And that's what these 9 are. So aseptic technique, calculations and measurement. And you'll notice with this training with Janet and Brittany, and in any of the other trainings that we do, we attempt to address all of these at least once. So we'll talk about aseptic technique. Y'all did that lab with Labster. We absolutely are addressing measurement, haven't gotten to calculations at this point. Reagent and media preparation, lab sampling, so the procedures for gathering samples and following through with that. Equipment use and maintenance is specific to the equipment that you're using, but to understand that each type of equipment has a particular way that it needs to be handled and also maintained, understanding the value of it. Record keeping and lab notebook. In our last training, we had a lot of feedback that people were wanting new ideas for that. And I love that Brittany just gave us a couple more ideas with the CER, with the color coding, and with group work on that. Inventory, it is huge. And so when you guys get your supplies in, for example, after this training, it's a really nice opportunity to have some of your students that are ready to take the inventory on the stuff that you get in. That's an actual skill that is actually
Cynthia's
needed.
raising her hand.
Oh, sorry. I
No,
can't-
you're fine. I was gonna wait till you got done because I had a comment about inventory. So this year, my kids, we set up their lab tables and it has all the equipment that they'll ever like primarily use. And they have a lab notebook and they have to check it before lab and after lab. And if they're missing supplies, they have to restock their stations. So
Perfect.
Nice.
That's that real life skill. And Felicia, do you want to share your story about your inventory? Putting you on the spot.
Yes, that's fine. I got some things from Georgia Bio, and I just went online and got a normal little inventory sheet, and I had the kids, I broke them up into their groups, and they all got certain amounts of the different materials that I had, and they just used this common document and did inventory for the lab. So now we know what we have in the lab, and we run out of something, they know what to tell me to order to get ready. It's very good. It's giving them opportunity to actually see the things that we reuse and to know about. I try to start telling them about the companies I order from now too. So
About some of the companies, what?
the companies that I use to order some things from, like BioRed and Fisher and all that.
Yeah. You're difficult to hear. You might have stopped talking, but basically, so I think that when Felicia's order came in from the Bioed Institute from the Equipment Depot, her students ended up doing some of the inventory there. So any chance really that you get to apply that's hopefully in a helpful way to you, gets that way eventually, is a win-win for everybody. I brought up lab safety earlier. That's a big one. And then following directions. So that falls into standard operating procedure. We're always trying to get the kids to follow directions, but that translates as you get older and into the professional world as standard operating procedure wherever you are. So these documents are in your folders in the Google link, and each of these skills are broken down a little bit more if you scroll down. And so each of these bullets are actual wording that Megan and I pulled out of job descriptions. And Indeed is a great resource. It's free. You can even send your students on there if you want them to try to find some jobs and careers that they may be interested one day, and we'll be probably surprised to find out how many they'll be qualified for upon graduation from high school, especially if you're in some of the career pathways. So I won't go through all of this, but just to let you know that these go into further detail throughout this document, and that some of the companies that we pulled from are listed as well. And these are all Georgia-based companies, and then what the positions are called. Any questions or comments? All right. Can I stop sharing? Where's my thing? I don't know. I don't know how to get it off.
It should be up at the top.
Yeah. It's not. Okay. Well, anyway, that about wraps things up. Janet and Brittany, did you guys have anything else for today?
No, that was it.
We think you guys should look at your tubes tomorrow and maybe take some really good pictures so you'll have a set of data as a reference. If you look at any of those and you're unsure what they mean, feel free to reach out to us, either email or do they have our phone numbers?
They have a group me.
Yeah, or send the group me, and we're happy to help. But yeah, just whatever you guys need. I know we're going through stuff really, really fast, but we're trying to just introduce you and give you enough to be deadly. And then just reach out to us for any support that we can provide. Kristen and Megan should be sharing a couple more folders with you that have tons of stuff in there on Central Dogma and on just kind of like lab management and reporting. And, one of the things Kristen was talking about SOPs, I don't know about you guys, but if you have taught babies and then you have some kind of upper level science class, Lord, there's nothing better than a kid that walks in and actually knows how to read a protocol and do a lab without asking every single word, like, wait, what do I do now? Man, it is like whoever that teacher was before, you were like, what, all of the Starbucks I have for you, all of it.
Yeah. And so to access these files, and that one of the very first emails that you were sent, it was really long. It had the Google link in there.
And if you can't find something, just put it in the group meet. Or if there's something that you want and you're not sure if it exists, we can either tell you if it does already, or we can try to find something like it. So just reach out to us if you need anything. We really do love this, and we're sorry that technology is challenging. And we really wanted you on our campus talking to us all day for 2 days straight. But we're so glad that we were able to offer something, and we hope that you're getting something out of it. And like I said, if we're hitting things too quickly at any point, or if there's anything else that we can do for you, please reach out to us. We really want to help. We love teaching biotechnology, and we really love what it does for students.
Thanks, you guys. Well, have a fantastic night. And if you want office hours for anything, shoot Megan an email about what would be a good time for that. She had them Friday, but nobody needed it, which is totally fine. See, now my thing popped up. I am technologically challenged a bit, but it was gone.
Challenging you too, so.
Exactly. Thanks, Megan. All right, so let Megan know if you want any extra help on that, and she's happy to set some hours or work out something with you. But have an awesome night.
Thank you for the sweet comment, Felicia. And Jackie, in the document that Kristen shared, there is more stuff there about quality assurance that you could probably pretty easily turn into some sort of, if not full on lab, definitely experience. Or you could create a lab where they're designed to not be able to do, and then they can have to troubleshoot through it.
Why did it fail?
Yeah. Why did you fail at science? All right. Thanks, guys.
Thank
and
you,
Megan
guys.
and Kristen, thank you for the cookies. You guys are fantastic.
Oh,
Thank you.
You're welcome.
Brittany and I next week may not be on the call. The folks from NEPCR are going to be doing the food safety lab, which is phenomenal. But I've done it multiple times, and if you have any questions about it, please feel free to reach out. Next Monday is actually one of our Jamboree days, and so we'll be off jamboreeing.
Yeah.
Except virtually.
We're gonna try to pop in, especially if you guys have questions or anything that you need, but we'll have all of our kids, and it's our one day we're gonna have our whole team together to try to enjoy something of Jamboree, because we should be in Boston right now with our kids.
Yeah. All right.
All right, guys, have a great week.
Good night. Thank you.
Night.
I'm just looking at the chat stuff.
I saved it.
Okay, awesome. I mean, usually when I download anyway, it saves them all separately, but
What do you mean separately?
When I download everything from the cloud, I get the video, the chat box.
Oh, yeah, yeah.
the script.
Okay, yeah. And then you can download it from there, so. All righty. And bye.
Did you see, Melinda Chansey's? She said, "You have our address, send the wine and cheese."
If we could.
I know, maybe there's some bourbon left from Summit. The biotech career site, is that, oh, okay, so that is not from Innovate. Is it from Innovate?
It is, but it's their, like, I think their last iteration before they were called Innovate. So like the BioLink or something. But
How do I find out more basic information about QA? Something I can change into a lab. I wonder if that's something we'd want to create a lab for.
Mhm.
I mean, it seems like, let me stop recording. It seems like we could